Good evening, everyone. Good evening, good evening, good evening. Here we are again. Sorry for the time delay. I just wanted to make sure that the questions I have selected for today's session are actually useful, and they are the ones that I want to talk about today. As you could see in today's title, tomorrow, not next week, not next year, tomorrow is finally our biology exam, which I'm sure that some of you were expecting it for a long time. Some of you are very scared, but the thing is, tomorrow we will try to do our very, very, very best for it. Wait a second, let me close Teams. Today there was a little bit of an incident with some interventions, but I believe tomorrow is going to be all sorted. Okay. How is the quality of the video? Is it good? Sound good? Everything good? Let me double check in my second screen. And in YouTube. Just to make sure that everything is fine. And then we can go to the questions we are going to talk about today in biology. I find hello Tariq, that's great. Biology has much more to study, much more to revise than physics, for example, but I would say that it's easier than chemistry. I think among the GCSE subjects, chemistry is probably the most difficult. Hello, how are you doing? Okay, so Twitch looks great. Let me see if YouTube also looks good. There are some people who prefer to watch the videos in YouTube. All right, where is it? Can anyone? Yes, apparently I see that. Yes, okay, cool. So I see that the YouTube also works well. All right, so no more wasting time. Let's go to the exam questions. And today we have a very, very busy evening. Let's set some music. I don't know if you guys, but I don't like working without music. I find it extremely boring, and when I'm very bored, I'm easily distracted. Probably is happening to some of you. All right, let's put some jazz. I love working with jazz. If anyone has any other suggestion for the music, you are more than welcome. Okay, but no reggaeton or anything weird. Okay, or rap. There's no point of a song with lyrics because that is going to distract you. All right, so here we are with my camera, the visualizer. Wonderful. Let me put the microphone a bit higher. Fantastic. So now I'm going to show you the first question I have selected for today's revision. It should appear in your screen in 5 seconds. There you go. Let me make it a bit larger. All right, can you see it well? A bit small. What if I put it? All right. I don't want to cover my face with it. Okay, so let's put it down in here. The first question, and I'm gonna use this because this allows me to talk about many different things, is compare prokaryotic and eukaryotic cells. Very basic. Topic 1. What is the difference between a prokaryotic and eukaryotic cell? Let's start with the name. Pro in Greek means before. And in some contexts in English, it will also mean pro. I'm trying to think like Nothing is coming to my mind now. Okay, hello. But pro means before, and the word karyo, Karyo, karyo means nucleus. So prokaryotic cells are the cells that they do not have nucleus, or they exist before the nucleus. Hello, Amil One Amar. Welcome to here. So prokaryotic cells, they are the cells that they do not have nucleus, or they exist before the nucleus. And EU means real. So eukaryotic cells, they are the ones that they have real nucleus. Just with the name, with a bit of Greek knowledge, we could find lots of things. But let's talk about the difference between prokaryotic and eukaryotic cells. And it's 4 marks. So if they ask you how many sentences you think you need to give to get the 4 marks, use your common sense. 4 sentences, minimum 4 sentences. And you should not spend more than 4 minutes in this question. Okay? All right. So, I think black is well seen. I will do like a table where I'm going to write down things that come to my mind, okay? And after that we can write sentences. So, prokaryotic, no nucleus. And when I mean no nucleus, it doesn't mean that they don't have DNA. They have DNA, but it is not surrounded by a membrane. Meanwhile, in here, we have nucleus. DNA in the cytoplasm. Or if you don't like in, you can say submerge. I like the word submerge, okay? So basically the DNA is just in the cytoplasm, just there, okay? Meanwhile, in here, the DNA, well, I said DNA, but it could be also RNA. Some bacteria, they don't have DNA, they have RNA. And in here, DNA is surrounded by a membrane. What other differences can we find between prokaryotic cells and eukaryotic cells? Do you remember how do we refer to the parts inside a cell? Let's say the organs of a cell. Anyone remembers? Anyone who would like to participate? Your local Olivia, those are examples of cells that they are prokaryotic and eukaryotic. Exactly. Tariq. Yes, Tariq. Just to let you know, both of them, they could have cell wall. Both of them. Both of them could have cell wall. However, what Waffles 5421 has said is exactly correct. So, prokaryotic, they don't have no organelles with membrane. And in here, we include lots of them. For example, organelle with membrane could be mitochondria, could be ribosomes there are. Mitochondria, Golgi, chloroplast, okay? Meanwhile, eukaryotic cells, organelles, surrounded by membrane, okay? And here you have the example I just mentioned, okay? Like mitochondria, chloroplast, Golgi, etc. Prokaryotic cells, they are simple, very simple. Meanwhile, eukaryotic cells, they are more complex, not complicated, okay? Not like Avril Lavigne song, like complicated, okay? You know the song I'm talking about. It's a very good song. It's a bit beginning of 2000, okay? Hello, Kai. Example of prokaryotic cells, I think they were mentioned earlier. We have bacteria. Meanwhile, example of eukaryotic cells, we have animal or plant cells. Let me put you a bit in a in a trouble. Let me put you in some difficulties. If I tell you protist or fungi, where would you put a protist or a fungi? Answering your question, prokaryotic, they are normally smaller. Normally smaller in terms of size. Hello, David. Yes, it is also foundation. It is also foundation. Very foundation. Please drive safe, okay? Where would you put protist or fungi? In prokaryotic or eukaryotic? Think that we refer to them later in the future when we talk about disease and pathogens. We talk about protists and fungi. So where would you put them? Prokaryotic or eukaryotic? What do you think? Come on, guys, it's much more fun if you participate. All right, I will stop your suffering. Protists and fungi, they are both example of eukaryotic cells. Eukaryotic cells, both of them. Okay? Yes, and protists as well. They are both eukaryotic cells. Okay? So the most famous example of prokaryotic will definitely be bacteria. Okay? That would be the most common one. Is there any other difference that you remember between prokaryotic and eukaryotic cells? There is one that I liked it. They are bacteria, they are normally, the prokaryotic cells, they are small or smaller. Meanwhile, the eukaryotes, they are normally larger. Okay? However, we have only talked about differences. Is there any similarity? Because the question is asking you to compare, so you could actually add some similarities. What they have in both? What can you find in both? We mentioned it earlier, RNA or DNA. Both of them are cells, so they have RNA and DNA. But there is at least two more similarities that you can take from here. At least two more. Yes, they both have ribosomes. Remember that ribosomes are needed for... No, they don't contain both of the mitochondria. They have ribosomes because ribosomes are needed for the synthesis of proteins, and both of them, they need proteins. Good. Exactly. Cell membrane. Those are three things that they both have in common. And cytoplasm. Love that. Yes. Well done. I forgot completely, but both of them have cytoplasm. Exactly. Exactly. Well done. Outstanding. So, using this information, look how many we have written. We have written a lot for one single question. Using this information, sorry, I will put... Using this information, can't you write 4 bullet points? Easy peasy. Hello, Tic Tac Gobbler. I'm sure you can find 4 bullet points from here. 4 bullet points. You can take maybe 2 similarities, 2 differences. 4 marks. Easy peasy, lemon squeezy. Can we go to the next question? Give me a yes, give me a no, give me a thumb up. Am I going too quick? Tell me something about it. Yes, wait a second. I think that the last part of the paper is not visible. Bloop. Now it should be visible, okay? There you go. 4 similarities and so many differences. Yes, this is one prediction. This is coming from a predicted paper, okay? So they always talk about cells. prokaryotic and eukaryotic. And they always talk about plant and animal cells. Always. That is something that is always there. Always. Because it's the base of cell biology. If you don't know the difference between plant and animal cells or prokaryotics and eukaryotics, then how are you going to talk about pathogens? How are you going to talk about mitosis? How are you going to talk about meiosis? How are you going to talk about specialized cells? Impossible. That's why this is the base. This is the base of your pyramid. You need to be able to understand differences between basic cells. By the way, brain. Which one do you think appeared on Earth earlier? Prokaryotic cells or eukaryotic cells? Which one do you think they have been here for longer? Prokaryotic. All the cells, they started as prokaryotic and they evolved to eukaryotic. Exactly. They evolved to eukaryotic by absorbing other bacteria. Absorbing other bacteria. Getting more complex. That's the trick. That's the magic. Let's go to the next question. No, eukaryotic is more modern. David, you are made of eukaryotic cells and you are complex. Bacteria, they are made of prokaryotic and they are more simple. Okay? Let's go with question 2, guys. Question 2. It will appear on your screen in 10 seconds. I say 10 and it's going to be 5. Bloop. All right. No, there will be a question about immune system, but specifically about phagocytosis, I don't think so. All right, all cells contain ribosomes. Describe the role of ribosomes. Super important. So, ribosomes, as we have just discussed, they are in both prokaryotic and eukaryotic. I think the required practical is going to be about amylase. The one about amylase. Protein synthesis. Good. So ribosomes, they participate and they are fundamental in protein synthesis. But I want to take a bit more from that question. So, what is the function of proteins in our cells or in our body? Whatever you prefer. I don't mind. It's a bit blur. Now it seems. Why do our cells need proteins? It's because they need to go to the gym and they need to get strong. I think the one about amylase. That is in our body. But let's think about cells. Yes, we need the proteins to grow and repair. Obviously, when you were born, you were this size. A baby is like this size. And now look how tall you are. Yes, we need proteins for growth and repair. But let's think in cells perspective. Why do they need proteins? Okay, so let me tell you, the proteins, they are, that's very creative point and not fully wrong. So proteins, the cells, they use them to create their own structures, okay? Like the structures of the cell. And also because proteins, they work as a chemical messenger. So the cell is able to do all their basic functions. We don't say real names in here, okay? So the cells, they use those proteins to make their basic functions, all right? The proteins, they are like the messenger. Like I am talking to you, the proteins, they move around the cell and they're the chemical messenger. And as someone said earlier, proteins, they are made of amino acids. But let me ask you one thing. From where does the ribosome get the information to assemble the proteins? From where? Where is it reading it? How do the ribosomes know how to make proteins? Yes, proteins are cell structures, that's why they need the proteins, and the cell function, correct, both of them. But how do the ribosomes have to make the proteins? Where did they learn it? Did they go to a course? Did they go to a school? Where did they go to learn that? I think someone got it there. Someone got it there. So, the instructions comes from the DNA, which is in the nucleus. It's in the nucleus, okay? But in the nucleus, it's too important to leave the nucleus, okay? So the DNA is always in the nucleus, and the DNA cannot leave the nucleus. So what we do is we have our RNA copy or transfer that is gonna go enter in the nucleus and photocopy the DNA. Then once the copy is done, it will go to the ribosomes, and the ribosomes will read the photocopy and make the amino acids. I know I'm simplifying a lot, but I think that this needs to be for all of you, foundation included, separate science for everyone, okay? So what I'm trying to explain is that the ribosomes, they get the information. They are in both prokaryotic and eukaryotic. So they get the information to do the proteins from the DNA, but not from the DNA directly, but from a photocopy of the DNA. Understood? I think your local Olivia, plant-related questions will be specialized cells, which we are going to go there in a second. So, ribosomes, the function is protein synthesis, and protein synthesis all happen thanks to a mechanism where another RNA, because ribosomes is a type of RNA, is doing a photocopy of the DNA. Anyway, let's go to another question. Yes, you have to, especially the xylem and the phloem. You need to know that. Let's go with another question. Bloop. Following question will appear shortly. We have lots of people in here today. Are you afraid of your exam tomorrow? Yo. Don't worry, guys. We all have lots of faith in you. Okay. This question is quite juicy. Look at this. It's for 5 marks. That you like, the one you hate. All of them. Guys, you will smash the exam. Look at this. This one is quite interesting question. So, a sperm cell is an example of a specialized cell. Outline and explain the features of a sperm cell and how they make the sperm cell adapted to its function. I have no clue what you are talking about. You all say it's come. Okay. Now I understand it. Hello, Cams. Happy to see you in here. Let's talk about the specialized cells. First of all, they are saying that the sperm cell, they are specialized cells. This question is gonna be in the exam, 95% sure. 95% sure. Specialized cells. Maybe not, they will not talk about the sperm cell, but they will talk about the specialized cells. I guarantee, I'm happy to bet money for this. There will be specialized cells. But let's do one step backwards. What is specialized cell? I know that you are all thinking on the answer. I'm thinking about the process to reach the answer. What is a specialized cell? What is a specialized cell? How the specialized cell is different from other type of cells. Okay? Do you remember this song? Last Christmas I gave you my heart, but the very next day. How is the sperm cell special, or what is the meaning of a specialized cell? Let me tell you two keywords, or two key sentences, better said. Specialized cells, they have a particular structure, or if you don't like the word structure, you can say features. But why do they look different? Because they have a particular function. That's the key, guys. Okay? The sperm cell doesn't look different because of the sake of looking different. It's because they have a particular function that other cells they don't have. That's the idea. If you think about in professions that you can see around you, if you think about a policeman or a policewoman or a doctor or a nurse, they all have different uniforms. Why? Each of them, they have a uniform that helps them to do their job. Specialized cells is the same idea, is the same metaphor. All they look different and they have different structure because they carry on a particular function. That's the magic. How do you know if a cell is specialized? Good question. What is the opposite of a specialized cell? What is the opposite? How do we call a cell that is not specialized? Do we call it normal cell? Do we call it boring cell? Do we call it friend zone cell? Do we call it I love you just as a friend cell? No. No. We call it stem cell. Stem cell. It's a cell that is not special yet. Okay? So stem cells, they don't have a particular function yet. And they're just there waiting to be used. For example, now you go to the kitchen and you help to cook the dinner, and I will talk about the meristem in a second. And you cut your finger, you start bleeding, you will lose blood, you will lose red blood cells, and then your body will start making new red blood cells. From where? From your stem cells. The meristem is a structure in plants that they pass from specialized to unspecialized back to specialized. Let me clarify this, okay? Look, our stem cells, once they get their job, once they get their function, once they get the specialization, they can't go back. So for example, if you have a skin cell, that will always be a skin cell. If you have a bone cell, it will always be a bone cell. If you have a sperm cell, it will always be a sperm cell. They cannot turn back the process of being specialized. However, plants, they can. If you get a flower, you cut it, and you replant that part of the flower. That part that you have cut can turn back to unspecialized again, and make roots, for example. Okay? Stop. We are going too quick. Let's talk about the sperm cell, which was the question, okay? So, now that we know what is a specialized cell, let's think about what about the sperm cell is special, okay? Sperm cell looks like something like this, okay? There are at least 1, 2, 3 particular or 3 elements of sperm cells that you could mention. You could mention about sperm cell. Number 1, how do we call the tail? Don't call it tail. Tail is not biologically accurate. It has another name. What is the name of the tail? tail? Come on, look for it. It's a name in Latin. Flagellum, exactly. You can also say flagella. Doesn't matter. Okay? Question, if you say tail, are they going to remove the mark? No. They are not going to take marks from you because you call it tail. However, if we could use a scientific term, why not? And what is the function of the tail or flagellum? To swim. The sperm cells, they need to swim. From where? From where do they swim? They swim from, let me see if I can make a diagram that is not inappropriate. So this is the cervix. This is the uterus. And then in here, the uterus divided. Okay? Something like that. Okay? These two are the ovaries. These are the fallopian tubes. Fallopian tubes or oviducts. Okay? If you prefer. And the egg cell can be found around there. We're going to find the egg cell there. Okay? When there is an egg cell that is mature, it will wait for the sperm cell there. So the sperm cell needs to go from the cervix, which is at the ends of the vagina. The vagina is in here. So the sperm cell needs to go all the way up to there. Which you might be thinking, that's a short distance. But the sperm cell, it takes 48 hours, 72 hours. More than that, the sperm cell dies, okay? But basically, to move from there to there, the sperm needs to swim. By the way, the flagella doesn't move like this. It moves making spirals, just to let you know. But that's not the only thing. What other thing we have? The sperm cell is full of mitochondria. Why there are so many mitochondria in the sperm cell? Why does the sperm cell need lots of mitochondria? Because in the mitochondria, there is a chemical reaction happening, which is respiration. Respiration. And why do we need respiration? Because in respiration is how the cells get the energy. And believe me, the sperm cell needs lots of energy. Lots of energy. That's why all those mitochondria. More things. The sperm cell contains DNA. DNA needed to fuse with the DNA of the egg. And also, in the head of the sperm cell, there is an acid. An acid on the head of the sperm cell to enter through the cell membrane of the egg cell. Yes, you can. You can draw, and you can use the drawing to explain it. So think that the sperm cell is not like going to the egg cell and doing just like this. No, it needs to pass through. That's why the acid in there breaks down the cell membrane and then enters. Why did it get? Sorry, because I moved the camera. Apologies. Do you see well? Alright. So you can see an egg cell. You can see an egg cell. You cannot see a sperm cell. A sperm cell, no. But egg cell, you can see it naked eye. It's the only human cell that you can see naked eye. Okay? By the way, the head of the sperm cell is called acrosome. But I think that's a bit too much. Okay? That's a bit too much. And if you notice, the sperm cell has a shape There's like a streamline head. Okay? Streamline. That is to reduce to the minimum possible the resistance, the friction. Okay? Streamline head. Okay? So it is easier to swim. Are there any parasites that can affect sperm cells? Yes. Yes. There are. It's not common because you make millions of new sperm cells every day. A healthy adult can make up to 40 million sperm cells, or can have 40 million sperm cells. Okay? Alright, this question has lots of things to say. Really lots of things to say. In my case, it helps me to draw the sperm cell, and by drawing it, I can start taking things out. I would recommend you to do the same thing. Alright? That's one advice I can give to you. Let's go to another question. Am I going too quick? No. Well, an embryo is when you combine an egg cell and a sperm cell. Okay, let me clarify that 1 second because people were asking earlier. So, the sperm cell plus egg cell, that makes an embryo. Okay? And what you are a bit confusing with what we talked earlier about the stem cells, there are two types of stem cells we can find, okay? Adult stem cell or embryonic stem cells. Remember that the definition of a stem cell is a cell that is not specialized yet. So it could turn into any type of cell. That's the magic of the stem cells, okay? They can turn into any type of cell. That's the magic. However, here comes the magic. These ones, the adult stem cells, can be found in bone marrow of long bones. In what bone mostly? This one. The one that goes from your hip to the knee. This is called the femur, okay? And I don't know if you have ever donated bone marrow because people have leukemia, they need stem cells, and I have donated stem cells, and it's quite painful. It's very, very painful because they put a massive needle inside your bone and they put it through your leg, all right? However, the problem with these adult stem cells is that they are multi-potential. What is the meaning of that? That they can turn into many things, but not into everything. For example, your adult stem cells, they can turn into blood. They can turn into bone. They can turn into muscles. They can turn into skin. They can turn into stomach. They can turn into liver. However, your adult stem cells, which you have them inside your body, inside your bones, they cannot turn into neurons, for example. And remember that neurons, they are for example, the brain cells. They cannot. So if you lose any brain cell, that's not coming back. Heart muscle cell. Remember that your heart is a muscle. If they get injured, your heart is not able to replace those muscle cells. Okay? So your cells, your stem cells, they can turn into many things, but not into all of them. However, the embryonic stem cells, that by its own name you can imagine, they are being found in the embryo, they can turn into any type of cell. What is the problem? What is the problem with embryo stem cells? Ethical issues. If you use those embryo stem cells, you are removing the possibility of a new individual. Think that a human embryo, after 9 months in the uterus, is a human. It's a new human. So if you are taking those stem cells, there's no human. That's why you have that ethical issues, religious issue about the usage of embryo stem cells. Okay? And there is like some sort of biological debate. For example, would you use embryo stem cell to repair the spinal cord of someone who had an accident, for example? Or would you use embryo stem cell to heal someone who has dementia or Alzheimer? Would you use them? That's a debate. That's a biological, ethical debate that is worth it to have in a different time. But that is the reason, okay? Embryo stem cells, they can turn into any sort of cell. Adult stem cells, they can turn into some. Okay? Healing is a no-brainer. What do you mean with that? Talking about a child being able to produce a sperm cell, that happens during puberty. In males, normally it happens between 13, 14, 15. Some never get mature. Okay? Was therapeutic cloning. All right. Therapeutic cloning. Let's talk about how does embryos normally do not get rejected, and I think nerve cells can't be. Yes, as I mentioned earlier, your brain cells cannot be replaced. Okay? Let's talk about therapeutic cloning before Bold Malteser gets absolutely crazy. All right. Let's start talking about how we are doing cloning, okay? First, cloning is not something that we are doing all the time or you can make easily, okay? It's not like that. I'm sure you all have heard about a sheep that was called Dolly. Have you ever heard about Dolly? Dolly the sheep? That was the first animal ever cloned. Yes, cloning humans is illegal. Cloning human is illegal, and you cannot actually, in general, research with humans is quite, I would say, restrict. Okay? It is quite restrict. All right. So, let me explain to you how is the cloning, okay? So, why is it illegal? Why do you want to make our new humans in a laboratory? How would you use them? How would you use them? We are not, guys, we are not God, okay? Okay, so we start with human egg cell from a donor, okay? This is a human egg cell. We're gonna call this from patient A. From patient A. Patient All right? First, we need to remove the nucleus. Question for you guys. Inside an egg cell, a human egg cell, how many chromosomes? How many chromosomes in an egg cell? No, 23. And a human has 46 or 47, if we are talking about Down syndrome, okay? So an egg cell cannot make a human, starting point. But if we remove the nucleus, then we're going to have an egg cell with no nucleus, and we're going to have the 23 chromosomes nucleus, okay? Or you prefer with half of the human DNA. Now we're going to get, imagine that we're trying to clonate Mr. Romero's left ankle. Imagine we want to clonate Mr. Romero's left ankle. We're going to get patient B. And from patient B, we're going to get a skin cell. But here comes the magic. The skin cell of patient B, of Mr. Romero's ankle, now this cell has 46 chromosomes. This skin cell has enough information to make a full human. Not half of the DNA. Your skin cells, only with one of your skin cells, I can get all your DNA code. So now I get this nucleus, and I will put this nucleus with 46 chromosomes inside the egg cell that was empty. So now I have one egg cell with 46 chromosomes. Egg cell with 46 chromosomes. If I leave now this egg cell with 46 chromosomes inside the uterus of a woman for 9 months, now this is going to be your clone. Okay, you don't understand. Give me a second. Let me see if I can use boxes, okay? Imagine this box is an egg cell. Normally, egg cells, they don't have all the instructions to make a human. They only have half of the instructions. Remember that to make you, your cells, they have the DNA of your mom and the DNA of your dad. Okay? Obviously, it's already 8:30. All your cells, they have both the information. Mom DNA, dad DNA. But your one egg cell has only one. Only one. This is not enough to make a human. I only have the instruction of one of them. This is not a human. This is an egg cell or a sperm cell. So what do? I get the egg cell. I have the egg cell of another person, not you. And I empty. So now I have the box with no DNA inside. There's no DNA inside. Okay? But imagine I want to clone myself. So now I go to my skin cell, take the nucleus of my skin cell out, and now I put my DNA in the box. Remember that the egg cell was empty before because I removed the nucleus. If I leave now this box, this egg cell, for 9 months in a uterus, what is going to be born is going to be a baby which is identically to me. Is identical to me. Why? Because the DNA I put inside here is a DNA that comes from my skin cell. So if I leave this inside a female for 9 months, the result is going to be a baby that is my clone. My clone. But we don't reach to that point. We don't go to the end point to make clones. We stop. So once we have the egg cell with my skin cell DNA inside, or hair cell DNA inside, or whatever DNA but is fully complete, not just the half of it, now we force this embryo to divide through mitosis, okay? Mitosis is division. And now instead of one single cell, it's going to make an embryo. But it's an embryo of my clone. It is not mixed with the egg cell, so it is going to be... No, because I am removing the egg cell. The egg cell, the first egg cell, I am removing the nucleus. So it's not going to be a mix of both. Because this nucleus, the original one, gone. Ciao, ciao. Sayonara. I don't need it. See you in another life. Gone. So what I have is an egg cell empty with no DNA inside. And then I fill that egg cell with my DNA. Anyway, so once I have that DNA, that egg cell with my DNA forms an embryo. Then I go to that embryo, which is my clone. It is my clone. So it is my DNA, my stem cells. And I remove those stem cells out. And I use those stem cells. Why? Well, some of you might be thinking, but sir, if you leave this embryo 9 months, it will also be a human. Yes, it will be my clone. It will be identical to me. That's why we will never do that. Never. Illegal, impossible, we will never do that. But we are using stem cells with DNA that is my DNA. So there will never be rejection. Okay? This is therapeutic cloning. This is therapeutic cloning. And this is not something very common. Okay? This is not something very common. Alright guys. Has been almost an hour. I have not gone to judo to be here with you. I hope you can appreciate it. No judo today, so I could help you with biology. Broke my heart. But I hope it's worth it, and tomorrow you actually have a very nice exam. Okay? I wish you the very, very best. If you have any question before the exam and you would like to share it with me, please do it. Sir, could you not use a clone to heal a broken part of your body? You could. However, cloning is a very delicate topic. Okay? It's a very delicate topic. And it's an area of science that we should not really get into. Otherwise, you will start having people making clones and use them as slaves. And probably it could be a problem. I recommend you to watch a movie which is called Once You Finish With Your Exam, okay? Now, no. You cannot do it now. The Iceland. This movie, as I mentioned earlier, my prediction is that the required practice is going to be the one about the amylase. Okay? This is a movie that I recommend you to watch. Maybe with your parents, okay? But it's about cloning humans and the potential hazards that we could have by cloning humans, okay? In this case, they clone humans and they use them as like a organ farm. Okay? And that's not very nice, obviously, because you are killing humans just to take their organs. So it's not very nice. All right? Very interesting topic. I don't think tomorrow is going to be therapeutic cloning in the exam anyway, because it was last year. All right? It was last year, so I don't think 2 years they're gonna talk about therapeutic cloning. If I were you, I would use my time left to study stem cells, disease, like communicable disease, respiration, and photosynthesis. Okay? With the time that you have left, is what you should do. Tomorrow, key stage 3. Yes, tomorrow is Tuesday. No, tomorrow I have tickets for the theater in London. So I would not be in here. Okay, so I will see you back on Wednesday. And Wednesday could be key stage 3. And on Thursday, I will try to do key stage 5. And then on Friday, I will do chemistry, paper 1. Okay? About microscopy or photosynthesis practical. The photosynthesis practical, I mean, if the photosynthesis practical appears in the exam, you just stand up and scream because it's going to be, it is an easy practical. Basically, you have your lamp, you have the plant, you move the light closer or farther, and you count the number of bubbles, and that's going to give you the rate of photosynthesis. Anyway, if you don't remember a practical, go to the website PHET. PHET. And there you have lots of simulations for you to work with them. You have for biology, for chemistry, and physics. Guys, it has been an absolute pleasure spending this hour with you. Now we'll have a shower and I will watch Vinland Saga before sleeping. Is it that fun and leaf on far away to see the weight loss of the leaf? What? 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 Yes, I'm in the second season now. God bless you all. Have a lovely evening. Ciao, ciao.